effects in oysters.
So I'm Kirsten Adams. I am an undergrad at LSU, and I'm studying the Genome-Environment associations in Eastern oysters in the northern Gulf of Mexico. Eastern oysters are very important to Louisiana's ecosystem and fisheries. They provide thousands of jobs, millions of dollars to the annual economy, and contribute about 23% to fisheries industry in Louisiana. But of course, that's not everything. We know that they have a large impact on habitat for a lot of other organisms, and they provide a good nursery for other animals such as blue crabs. They improve water quality with one oyster filtering about 50 gallons of water a day, and they provide good shoreline protection, just like the mangroves in South Florida, protecting from erosion and storm surges. So why are we studying adaptation to salinity? We know that oysters have an effect of lower salinity leads to lower growth rates and higher mortality rates, and high salinity leads to more predation and higher disease rates. We see this in a study that was also done in our lab, where they took salinity on the x-axis by condition index, and condition index is basically a ratio of wet tissue to shell size. So a higher condition index is basically a very fat oyster, and a lower condition index is a skinny oyster. And basically what this study and other studies found was that there is a good difference between how low and high salinity are seen in oysters, and we can actually see that in this photo. This is the same clutch of oysters, but these on the left were grown at high salinity, and these on the right were grown at low salinity. So you can really see the difference in growth and health between those. And if you look on this map, this all of this water is the Mississippi watershed that goes out on Louisiana's coast. So we have a very big salinity gradient along the coast. And, that's not the only thing. Our own coastal restoration projects also lower salinity. We have, in Louisiana, we are taught from the time that we enter elementary school that we lose about a football field of land a day to water. And to combat our land loss, we have the Coastal Restoration and Protection Agency, which implements sediment and freshwater diversion plans. But those plans also increase freshwater and lower salinity, which has a detrimental effect on our oyster populations. But it is important to note that we need our coast because that is where people live, and we love our people, so we don't want to lose that land. So my project addresses the question, what genes are responsible for increased salinity tolerance in Castrostrea virginica? And I want to familiarize y'all with this map. So these are all of the populations that were collected by my lab, and these 5 up here, Aransas Bay, Calcasieu Lake, Marsh Island, Sister Lake, and Lake Fortuna, are the 5 that I'm working with. And we have a general salinity gradient of increasing salinity as you move from East Louisiana to Texas. And then in this salinity map also shows salinity on the y-axis and is arranged in the same order. And we have sort of an upside down bell curve as you move along that salinity gradient. And for the methods, I took the low coverage whole genome sequences from those 5 populations, about 30 oysters per population, and I use ANGSD to get the covariance matrices and SNPs in those populations, and I use the covariance matrices in R to get the eigenvalues and the vectors of those eigenvalues to make a PCA, and then I use SAM tools and BCF tools to make file conversions that no one else cares about. And I got this PCA, which shows the genetic differences between populations. So we have Calcasieu Lake on the left, Lake Fortuna, Aransas Bay, and then we have these 2 overlapping, Marsh Island and Sister Lake. And it's really interesting because if you look at all of these together, you'll see that our middle population is actually our West Texas population, And then we have the opposite sides of Louisiana over here, and these two are the populations that are closest together. So it is very possible that it is just restrictive selection for the location of those, since that is only 60 miles apart. And then these are about 100 miles apart. Marshall and Calcasieu Lake are about 100 miles apart, and obviously Aransas Bay and Calcasieu Lake are very far apart. So that is very much a possibility, but considering the fact that the PCA is not arranged geographically, we should be able to find those genes responsible for salinity tolerance, since it's not completely confounded between distance and populations. We are currently trying to run the Genome-Environment associations. This is another result from our lab where they found that the salinity and this is, the circles are 6 parts per thousand salinity and the squares are 12 parts per thousand salinity, and we can see that they are only grouping based off population. And this was a transcriptomics study that was mainly on gene expression, so we know that it's much deeper than just on a gene expression and salinity tolerance level, which is why we're looking deeper into the genome at what the other genes would be. And to do those genome-environment associations, I'm trying to use BioC Manager and LEA, but I've run into some issues with file conversions and imputing, so if you have experience using these, please contact me later. So key takeaways is oysters are important, but I probably don't need to tell you that because this is a marine ecology conference. Low salinity has a detrimental effect on oyster growth and health, and we have many sources of fresh water on Louisiana's coast, so it is important to find out what those are, what those genes are, so that we can target the parental stocks for oyster restoration. And we know that there are population differences, and we plan to investigate those with Genome-Environment associations. So that's all. Thank you.